So Philippe got his BC from University of McGill University in 1994, and he did his PhD finished his PhD in 2001 on ecological and evolutionary biology in Princeton. And he was appointed in the Department of Earth and Ocean Sciences just 1 year before myself. So we kind of started more or less together with this Philippe. And what I want to say a little bit about Philippe is that his interests really lie in microbe world. However, when he was doing his PhD studies, he has done quite seminal papers on the effects of the carbon dioxide on the diatoms and calcifying organisms, physiological calcifying organisms. However, what Philippe is doing these days is his interest is kind of widening up and actually narrowing down, because he is really developing and capitalizing on a new equipment which allows you to do continuous, very tiny and very precise measurements of certain parameters like gases of the interactions. And at the same time, he's like kind of zooming into the very small scale, but at the same time he's going in another direction, going into the bigger scale, trying to use satellite-derived information and how it's actually going to help to understand what is going on underneath. So without actually going into many details, and I hope Philippe will be talking today about this a lot, I'm presenting Philippe.
I actually start my day, almost every day, in this building. I ride my bike in and I take my shower and I put my stuff in a fisheries locker. And I've always felt guilty about taking a fisheries locker, so I felt that this was a small step towards rectifying that. But more seriously, in May of this year, I went to the Pacific Biological Station in Nanaimo for the first time in my 17 or 18 years in BC. I worked closely with the Institute of Ocean Sciences, which is in Sydney, near Victoria, which is where a lot of the oceanography happens, whereas the fish people, quote unquote, are in Nanaimo. And I'd never been there, and that seemed sort of symptomatic of the gulf that sometimes exists between lower level, trophic level stuff and physical, chemical oceanography and fisheries research. So I went to Nanaimo. I felt really proud of myself that I had bridged this gap. And then they said, "What do you talk to the fisheries people at UBC right across the road?" And I said, "Not really." So actually, I realized how long I've been too isolated, I think, from this community. So the reason I'm here now is hopefully to begin some discussions and to give you some idea of what we're doing just across the road, so that we can follow up much more informally. It's a real tragedy, I think, that I haven't done that more in the past. So I'll apologize and hopefully start to make amends. I'm going to talk now as an illustration of the work that we've been doing around an NSWIC strategic project that's just winding down. It involves many, many people at UBC and elsewhere, including a number of fantastic students and postdocs whose names are underlined. Oh my God, Brian, I'm so sorry. I'm so sorry. Apologize. So Brian Hunt and Hakai Foundation is here, DFO is here, and many others who are probably also not there. My God, that's okay, there we go, first slide. So my, what do I have to point this at? This? Yeah, so my goal...
You just pressed.
Yeah, I just pressed. So my goal, as I said, is to describe some of the work that we've done, the development of some new tools that allow us to observe the ocean in a particular way, with a focus, for the time being, on phytoplankton biomass productivity. But I'll also talk about the work that some of our colleagues have done looking at zooplankton, this critical second link in the food chain. And I'll talk about also the application of these methods to remote sensing. The hope is that we can use these new sources of information and these new technologies to better understand ways of predicting variability in fishery stocks. And finally, as I said, my goal here today is to provoke some conversations with many of you here. So my overarching question then is to understand how variability in the physical and chemical environment of the ocean drives variability in ecosystem processes. This is just one example of many of the sorts of variability we see in ocean environments. This was the so-called blob, this warm water anomaly that came through in about 2014 and had significant implications downstream on the ecosystem. There are others, including ocean deoxygenation, which is something I'm working on now with Rashid, acidification, changes in stratification, nutrients, etc., etc. The ocean is subject to all of these perturbations, and it seems quite obvious that these changes in the physical environment are then going to impact the lowest levels of the ecosystem down here, which is what I study primarily, and therefore have ramifications up to higher levels of the food chain until we have things that are directly relevant to those who study fisheries. The challenge, though, of answering these questions about physical and chemical perturbations of an ecosystem is fundamentally one of scale. The ocean is big, as you know, and things are different across vastly different time and space scales. So how do we actually map these physical, chemical perturbations and ecosystem responses on anything that's like an ecologically relevant timescale? The traditional approach in oceanography was to go out on a ship and collect seawater in bottles, pull the samples onto a ship, maybe ship them back to a laboratory, and make many, many analyses of all kinds of things, whether it was nitrate or chlorofluorocarbons. We would have these particular sampling stations here, like at Ocean Station Papa, which has been sampled since about the 1950s. It's one of the longest near continuous ocean observing sites anywhere in the world. And we've been measuring, as you can see here, the concentrations of nitrate for 6 or 7 decades at this point. And this kind of information gives us an interesting long-term perspective, relatively long-term perspective. The problem, however, is that it's still fundamentally a dot on a map. So the combination of these kinds of efforts led in the 1980s and 1990s to these kinds of maps. This is something called WOCE, the World Ocean Circulation Experiment, that was based on exactly these kinds of bottle samplings. And by the end of the 1990s, this was the kind of map of the sections, the oceanographic sections, that had been completed as part of the WOCE program. And you can see the coverage is reasonably good, but A, there are gaps, and B, many of these lines were only completed once in time, so there's no repeat sampling and no temporal resolution. Things have moved pretty quickly in the last decade or two with the advent of a whole variety of autonomous sensors. This is the Argo program, which involves the deployment, literally the chucking over the side of these kinds of instrumented devices. Initially, they just had temperature, salinity, and depth sensors. Now they have the so-called bioargos with oxygen and a variety of optical sensors. And as of February 2018, which is the one I found easily on the web, this is the number of Argo floats that have been deployed. So there are these robots now floating throughout the oceans producing all of this information. Those are now being supplemented by gliders that are moving through a water column, resolving space and time. And one important point that I want to make to you is that all of these data are publicly available. And I actually teach a class next door for graduate students where we go through how you access these and many other sources of data to assimilate them into the work that you're doing. If you're working on a problem related to fisheries, you may have interest in extracting primary data on temperature, salinity, mixed layer depth, nutrients, all of these things. And so for those of you who are interested, I welcome you to come and talk to me about this course. It's sort of midstream at the moment, but perhaps for next year. Okay, so taking this approach of the Argo program and these autonomous sensors has really motivated the work that we've done for, as I said, almost getting on to 20 years now. And this is an image of the laboratory on our ship, the John P. Tully. It's a Coast Guard vessel. And this is the sink. The sink is connected to a seawater pump that draws water from about 5 to 7 meters below the water line, and it just distributes that water all throughout the laboratories. And this is one of the laboratories, the so-called wet lab, and all around here are different sensors through which this seawater is flowing as it's coming up from the ocean and then back down through the drains. I'll talk more about the details here, but what's shown, and I must say not very well, is these different optical sensors that we have. So I'd like to start, oh, and sorry, this is another example on a different ship. Now this is the Canadian Coast Guard ship Amundsen. This picture was just from a couple of months ago. This is a student of mine, Robert, working in the Arctic. This is more related to the work that we do measuring dissolved gases in seawater. And so I'll talk now for the next little while about optical measurements and gas measurements as a way to infer biological processes in the oceans. So in terms of optics, and this is not an area that I'm a specialist, but I want you to understand two fundamental things. When light, when photon enters into the surface ocean, photons enter into the surface ocean, there are two primary fates of those photons. They can either be absorbed into a molecule or a particle, or they can be scattered. And the scattering can be in the forward direction or in the back direction. So photons enter the ocean and they're either absorbed or they're scattered. And these absorption and scattering properties are what we call the inherent optical properties, or IOPs, of seawater. And for the next few minutes now, I'm going to describe how we can use these IOPs to derive very powerful information about the way the ocean works. So here's an example of what we would call an absorption spectrum for seawater. On the bottom, the x-axis is the wavelength of visible light from 400 to 700 nanometer PAR, photosynthetically active radiation. And on the y-axis here is the absorption due to particles. And we know that they're due to particles because we can run whole seawater and then we can run filtered blanks with all the particles removed. And by difference, we get the absorption characteristics due to particles. And these are not exactly matched to the location, but this is an illustration of what you would get in low productivity, offshore, very, very blue water, like what you'd see near Hawaii, for example. And you'd have an absorption spectrum that looks like this. When you put more particles in the water, both living particles like phytoplankton and non-living particles like dead organic materials, you have much higher absorption of these different wavelengths. In this particular case, you're absorbing a lot of the blue, you're absorbing a lot of the red, and the greens and yellows are less absorbed, which is why they're reflected back to your eye. So just the color changes in the oceans are reflective of changing absorption properties, both dissolved materials and particulates. It turns out that measuring what's called the absorption line height of this particular band here gives us a very good way to measure the concentration of chlorophyll. Chlorophyll, I think you probably know, is the primary photosynthetic pigment. It's used by all photosynthetic organisms, including those that inhabit the surface ocean. So if we measure with our optical approach, using this line height, the concentration of chlorophyll, we can calibrate that against the quote-unquote old-fashioned way that we used to measure chlorophyll by collecting seawater in a bottle and then putting it on a filter, extracting it in acetone, and doing all that kind of stuff. And we see that we have really good agreement. So we now have a way to measure chlorophyll. And I'm saying this as though it's relatively new. This actually isn't all that new. This has existed for a long time. We've just developed these capabilities here. And this is work done by Will Burt, who's sitting back there. So the chlorophyll measurements are not interesting particularly or impressive, but they are a good starting point. They're a good starting point, but they're not enough. Because just a measure of the pigment, the photosynthetic pigment in seawater, is not really going to tell us everything we need to know. Beyond the total biomass of phytoplankton, we need to know something about the size structure, how big the individual cells are, and the taxonomic composition. So I'm going to give you an example of why that matters. These are two hypothetical ocean ecosystems. This would be reflective of a high productivity, say like an upwelling regime off our coast here, where we have high nutrients that feed the accumulation of large diatoms, like chain-forming, large phytoplankton, like chain-forming diatoms. Those diatoms can be fed on directly by krill, which in turn can be fed on directly by whales or many, many fish. In contrast, when we go to those blue offshore waters, we have very, very low nutrient concentrations. Those tend to favor primary productivity dominated by very, very small cells, which in turn need to be eaten by progressively large zooplankton. So we have multiple steps in our food chain before we get to our top predators. And in terms of trophic transfers, if we have a short food chain, we're essentially going from primary productivity to fish in a single step. As the phytoplankton get smaller and smaller, we are introducing more steps and more potential to lose energy through respiration. So the difference between this scenario and that scenario is actually potentially as much as 2 orders of magnitude in terms of the amount of energy flow to the top predators. So how do we get an estimate of the size structure and the taxonomic composition? One of the ways it's been exploited in oceanography for quite a while is to exploit the fact that chlorophyll is not the only pigment that exists in these organisms. There's a whole suite of photosynthetic pigments, these so-called accessory pigments like chlorophyll C, chlorophyll B in some cases, and these carotenoids that account for the fact that phytoplankton can have a whole suite of different colors depending upon the complement of these pigments. And we can use the composition of these pigments essentially as a chemical taxonomy, because different groups of phytoplankton, like diatoms versus cyanobacteria versus haptophytes, have different characteristic suites of pigments. And this next table is going to be really overwhelming. I'm just sort of showing it to you for the purposes of a little bit of rigor. And that is simply to say that there is a lot of work that is not being done by us, but we've exploited it, that demonstrates how different groups of phytoplankton can be characterized to some extent, in many cases to a reasonably good extent, by a combination of these different pigments that we measure. So you can extract tens of different pigments from seawater and measure their concentrations and use these empirical algorithms to estimate the fraction of the community that is associated with diatoms or haptophytes or cyanobacteria. And then you can look for some empirical relationships that allow you to predict the abundances of these different groups. And again, there's perhaps more information here than you need to know, but I'll just tell you that these three panels represent a measure, an empirical measure of the size class of the phytoplankton, from the percentage that's in this pico-phytoplankton size class, which is up to, I think, 0.2 to 2 microns in size, so the really smallest cells. The nanoplankton are the 2 to 20 micron, again, very, very small, and the microplankton are all the plankton bigger than 20 microns. And what I'm showing you for both these size classes and these different taxonomic groups is an empirically derived relationship that we have used, and others before us, that relates the fraction of chlorophyll in these different groups to the total productivity of the ecosystem in terms of chlorophyll. So for example, I'll just use two examples here. In the microplankton, or the diatoms, which are these larger phytoplankton, as the amount of total chlorophyll in the system goes up, we have an increasing dominance of the phytoplankton assemblage by diatoms. Conversely, the picoplankton and the nanoplankton become less relatively abundant as total chlorophyll goes down. In the chlorophyll, we're shifting between these two kinds of environments. So high chlorophyll concentrations are a good indicator that we have large phytoplankton and dominance by diatoms. So because we can measure now chlorophyll from satellites, we have the capability to extend these sorts of empirical algorithms into the domain of remote sensing. So now all that we've done is we've taken a measurement of chlorophyll, not from a ship or from an optical sensor, but from a satellite. And we've just mapped this empirical algorithm on it to get this picture here, which represents the frequency or the relative abundance of diatoms in an assemblage as based on a chlorophyll image from a satellite. And we can see that the diatoms are very prevalent in these coastal waters, which happen also, of course, to be the highly productive waters with high chlorophyll. That's really just a function of the algorithm. Conversely, if we look at the abundance of other groups, like these very small picoplankton, here's an image of Senecacoccus, which is just a couple of microns or so, we see that these groups have highest abundances in the offshore low productivity waters. Okay, so these are just maps taken for September of 2016, which is when Sarah was playing around with this. We can then take that approach and look in different of these so-called Longhurst provinces. These are sort of biogeographic domains within the oceans defined by Alan Longhurst. And we can look at the abundance of different groups, whether it's the microplankton or the pico and the nanoplankton, in a time series. We can go back over the 18 or so years of the Aquamotus satellite, and we can say what happens, for example, in this province or this province to the abundance of microplankton or to the abundances of these other groups. And without really interpreting these patterns at all, we can see that there do seem to be some shifts, if you can believe this, in these different size classes in these different regions. So there's a lot more analysis that has to be done. We can look for signatures of El Nino events. We can look for a signature of the blob. But at least it's a way for us to begin asking questions about not just total chlorophyll, but the composition of that chlorophyll over time. So that's a potentially important tool. And I really want to emphasize that we're not the only group that's doing that. Brian has another project with my Sierra Costa at the University of Victoria. I think the focus, Brian, is mostly nearshore coastal waters. So this is really more of an offshore thing, and I think the two will complement each other. So this is not a unique project, but it's a capability that we've developed in-house for our particular area of interest. We need to move beyond chlorophyll, though, not only because it doesn't give us information about the taxonomy, but it also doesn't give us information about the productivity. And the reason that that's so is that when we look at the productivity of a system, P, we need to understand that that is derived as the product of two terms. One is the biomass, or the standing stock of material, and the other is an estimate of a turnover or a growth rate. If I show up in a location that has a high concentration of chlorophyll, it's possible that all the nutrients are exhausted and the turnover, the growth rate, is very, very low, so productivity is extremely low. And I would say that one of the biggest mistakes that many oceanography students do in the early years is to confuse productivity and biomass. So chlorophyll, at best, is an estimate of biomass. It will never give you, ever give you, on its own, an estimate of a growth rate. But as you'll see, there's a trick that we can play that allow it to point in that direction. So, as I say, chlorophyll doesn't provide a metric, a robust metric of biomass, for reasons we'll see in a moment, but just know now that the amount of chlorophyll in the cell changes all the time. Chlorophyll is regulated up and down with light and nutrients. So the stoichiometric quantity between chlorophyll and, say, carbon is really not a fixed value. So the reviewers will always get on your case if you say, we measured phytoplankton biomass as chlorophyll. They'll say, no, you measured chlorophyll. You didn't measure phytoplankton biomass. The other thing, as I said, is that chlorophyll doesn't provide a measure of turnover rates. So here's where we come back to these measurements of backscatter. Remember, backscatter is just photons that are scattered in this backward direction off of particles, off of the seawater itself. There are many constituents in water that will scatter. And just to remind you, if we measure the backscatter of its total seawater sample and we subtract a blank that's filtered, we're left with a particulate scattering signature. And our colleagues at Oregon State University and other places have demonstrated that there's a very good empirical relationship between backscatter, measured here, shown on the x-axis, and something here that we call phytoplankton carbon. So that's actually an estimate of the carbon that's associated with living phytoplankton, and it's actually a complicated method involving flow cytometry and sorting living material. And there's this relationship, and others like it, that show that backscatter is actually a very good proxy for the abundance of phytoplankton carbon. Which is to say that basically phytoplankton carbon is a dominant contributor to light backscatter in the ocean. Okay, so now that we have a way to measure chlorophyll, as we've discussed, and a way to measure carbon, we can put those two things together to measure the ratio of carbon to chlorophyll in these marine particulates. And again, this is work from Will Burt, and there is a distinctive variability from probably the lowest values, close to about 10, all the way up to 200 or more. And this variability doesn't appear to be random. There is spatial coherence. In other words, there's a zone here of elevated carbon to chlorophyll. There's another zone through here. So it doesn't appear to be a random distribution in space. And now what we're going to do is try to explore how we explain this and what information it gives us. So to understand why carbon to chlorophyll is useful, you need to consider how it can be used as a physiological proxy. And I think the key to understanding that is to consider the fact that carbon in all life is very much a structural material. Membranes are made of carbon, all proteins, carbohydrates, lipids, all these biomolecules have carbon. So carbon is sort of like the structural material. Of course, it's involved in catalysis. But chlorophyll is really just used to generate energy. This chlorophyll is a really small component of the total structure, the total nitrogen and carbon of a cell. But obviously it's playing a really key role in the harvesting of energy through photosynthesis, light-dependent photosynthesis. And so you can think about variability in the carbon to chlorophyll ratio as really an adjustment or a reallocation of light harvesting capability. So chlorophyll is really like the engine of metabolism, and carbon is kind of partly metabolism, but lots of other stuff that's just holding the whole thing together. And the analogy that I've often thought of is the difference between a minivan, and no offense to those who have minivans, no offense to those who have Formula One cars, but if you consider the ratio here of engine or energetic power to structure, clearly, in this case, we've got more engine, more energetic capacity per unit structure, as opposed to here. We've got a lot of kind of carbon, a lot of structural material relative to the engine. So this is my analogy for a high carbon to chlorophyll, and this is a low carbon to chlorophyll. So what is it then that drives variability in carbon and chlorophyll? Well, the first is light. If you give plants very low light conditions, they will upregulate the amount of chlorophyll so they can capture more of that light energy. Conversely, if you give them high light, in fact, in this case, it looks like excess light, which creates photo damage, but let's say sort of moderate light, the chlorophyll concentrations go down. So there is no doubt in both the terrestrial and the oceanic realms that the variability in carbon and chlorophyll is driven at first order by light variability. Low light, high chlorophyll, high light, low chlorophyll. That's not surprising and very, very well known. The question is, how do we use this variability to infer something quantitative about the system? So there are many, many approaches to this. The one that we've used, again, is from our collaborators at Oregon State University. This is an equation whose details don't really matter. I just want to point out that the ratio of carbon and chlorophyll predicted by what's called this photoacclimation model depends basically in this model only on light. And light, of course, is determined by the depth of the mixing, the day length, the surface irradiance, all these kinds of things. But it's a complicated looking, but ultimately pretty simple equation that just says light drives variability in carbon and chlorophyll. So, what does that look like in our terms? Now I've taken a little bit of that data that I've shown you in terms of carbon and chlorophyll, and I've plotted it out along an east-west section from near coastal waters out to the ocean station Papa in the middle of the Subarctic Pacific. The black dots here are our measured observations. The red is what we predict solely on the basis of light. We can get the mixed layer depth, we can get the surface irradiance, all these kinds of things. And we just apply this equation to predict a value of carbon and chlorophyll. Okay, that's all that we've done. And what you see over at least this part of the transect, the agreement is actually reasonably good. Which is to say that we can predict a lot of the variability in the carbon to chlorophyll ratio just from light-dependent photoacclimation. But then there are these other areas, particularly here, where our nutrients, this is mixed layer nitrate concentrations, go to zero, to these depleted values, where we have this excess structural material relative to energy, high carbon and chlorophyll. And in short, the answer to that appears to be that under these conditions of low nutrients, phytoplankton are just limited in their growth, they're not growing quickly, and therefore their energetic demands just go way down. They don't need as much light harvesting capacity when they're limited by nutrients. And this is entirely consistent with laboratory observations where we can grow cultures that are limited by nutrients, and the carbon to chlorophyll ratio scales exactly as expected with growth rates. So as we get them more nutrient limited and they grow more slowly, the carbon to chlorophyll goes up. And that high carbon to chlorophyll is consistent with what we're seeing here in these nutrient limited waters. So all of a sudden, the carbon to chlorophyll ratio is starting to give us some insight into the biology and the physiology of these organisms. So now to come back to this notion of primary productivity, which is productivity derived from a measure of phytoplankton biomass and a growth rate, I told you that we can derive phytoplankton carbon from optical backscatter. I told you that we can derive some information about phytoplankton growth rates from the ratio of carbon and chlorophyll. And when we multiply these two things together, we have a way to estimate productivity. So this is what our data look like. This is from the autonomous ship-based sensors. We're using optical backscatter to derive carbon. We're using the carbon to chlorophyll ratio to derive a growth rate. And this is what our productivity looks like. Okay, well that's fine, but is it right? Is it even reasonable? To test that, we used independent measurements with the old-fashioned, dating from the 1950s technique of carbon-14 experiments. So you take a sample of seawater, you put it in a bottle, you add carbon-14, CO2, you leave it in the light, and you measure the amount of that carbon-14 that's converted into organic material. This is a classic measure of net primary productivity. And please notice that this is a log-log scale that hides a lot of variability, but to first order, at least there is a good correspondence between these optical measures that we're deriving from a ship without ever touching a bottle, without ever touching seawater down here, as opposed to what we measure with a traditional method based on a bottle incubation. So now that there's a little bit of validation, we can go back to the satellites and say, what do we do with this? We know that we can measure chlorophyll from satellites, as we described. I haven't told you this, but there's also another product available from the satellites, which is a backscatter measurement. The satellites are also measuring backscatter at particular wavelengths. So when we take carbon and chlorophyll together, we can derive the carbon to chlorophyll ratio, which, as I've described a few moments ago, can be used to give us information about growth rate. And if we have a carbon measurement from a satellite product and we have a growth rate from a satellite product, we now have a way to at least derive an estimate of net primary productivity from satellites. And this is work that's really based on the pioneering efforts of Mike Barenfeld and others. And what we're doing here is adding to the confidence in that by validating it. So I want to make sure that we leave time, so I'm going to skip all of this. Just to say that we have other independent methods to look at productivity based on oxygen dynamics, and those are also giving us confidence in these other approaches. So just to summarize quickly before I move on to zooplankton for the last few minutes, I want to say that it appears from the work that we and others have done for a number of years now that these autonomous shipboard systems and these remote sensing systems are really yielding powerful information into large spatial and temporal patterns of phytoplankton biomass and phytoplankton productivity, really at the base of the marine food chain. And it seems to me that this information is going to be useful for those who are doing fisheries modeling and interested in assimilating information about lower level trophic dynamics to understand variability in fish stocks. And to take some step towards that, we're working with Sue Grant, who's at DFO Applied Science and is in charge, as I understand now, of the sockeye, the Fraser River sockeye assessment and predictions. But I imagine that there's many others here that are doing similar work, and I really would love to have some conversations with you. So even if we're feeling like we've got some progress towards measuring low-level plankton dynamics, we need, at some point, to make our way all the way up this food chain. So there's a lot of elements in here that are still unconstrained by what I've told you. And this is where we've continued to exploit these optical sensors in a number of ways and also to work through collaborations. So this is kind of a busy looking slide. But what I'm going to try to tell you is that these same optical measurements that we're using to measure phytoplankton carbon also turn out to be really useful to measure zooplankton. And in particular, what we're seeing is a signature of these diel-vertical migrations, which is to say, it's well known that many zooplankton come up into the surface layer to feed at night, and then they go back down at depth. This is what a traditional set of optical data would look like. So this is backscatter measured over the period of about 14 days, 2 weeks. The gray lines here are nighttime measurements. The way we traditionally handle these data is we just smooth them with the running mean filter. That's the way everyone does it. But Will, who had to go now, noticed when he was doing this that every time at night his data would start to get really noisy. He said, I don't know what's going on with the system. There must be bubbles in it. There must be something. It's getting really, really noisy. And so that noise is just reflected in these raw data as all these really high electrical spikes. It just looks like noise. It looks like bubbles. And to make a long story short, when he did a careful analysis of this, it turns out that these spikes would onset, in most cases, right around sunset, when the zooplankton were starting to come up. And he's done many other analyses looking at derivations of the size and all this sort of stuff that suggests that this is actually a really interesting way to look at the appearance of zooplankton in the surface layer. And that's something that he's continuing. I want to talk now briefly about some of the work that Liam Kwong is doing with Evgeny, trying to use discrete measurements of phytoplankton, plankton and other organisms across different size spectra, to use the size spectrum analysis to really infer the balance between growth and mortality. We have a high abundance of small organisms, a much lower abundance of large organisms, and the shape, the slope of that is really reflecting the recruitment of organisms across these, or the transfer, I guess, of energy really across these different size spectra. I know I'm butchering this, so maybe in questions Leanne or Evgeny can clarify. And this is really started by some zooplankton net sampling running through a laser optical plankton counter in the laboratory that's assessing both abundance and size of these different groups. This is a beautiful method because it can work on old samples. You can preserve samples and go back in time, and that's exactly what Leanne did. And what she did was to just look at the slope of this relationship from her data analyses going back, at this point, 20-odd years. And all I want you to see is that there's a lot of variability in the slope. In other words, the slope that she's deriving is bouncing back and forth. And if you take the slope as an index of a trophic transfer efficiency across these levels, it implies that the efficiency of energy transfer across these different trophic groups is highly variable. And I don't know what sort of models some of you do, but typically we don't allow that variability to fluctuate all the time. So that was a really important result I felt from Leanne's work. Another quick thing to talk about is an independent estimate of productivity of zooplankton that's being done at the University of Victoria by Teresa Vanello and John Dower. And this estimate is really focused on crustacean zooplankton. These are organisms that go through various molting cycles through their life. Here's a beautiful picture of a copepod shedding its exoskeleton, which is a strong component of chitin. And this shedding and molting involves the activity of an enzyme called Chitobiase. And Dower and others, based on earlier work, have used this Chitobiase assay. It's basically an enzyme assay to give information on the abundance and the molting cycle of these copepods. And without going into the details, some of which I don't understand, they have a way to estimate secondary production from this Chitobiase. So it's secondary production specifically of copepods. We have lots of primary productivity measurements, and when we combine those together, we have an independent estimate of this trophic transfer efficiency. How much of the phytoplankton productivity is getting transferred into the second trophic level? And these are results from Teresa's work showing that there's, as Leanne observed, significant variability in this trophic transfer efficiency. These are 2 years of data over different times of the year. This is the west coast of Vancouver Island. And I just want you to see that there is spatial and temporal variability in the size of these dots, which is corresponding to variability in the trophic transfer efficiency. Again, with significant implications for models. And so on the point of models is where I want to wrap up. I see that William's here. William is one of the collaborators on this project, and he's really leading our efforts to assimilate all of these new sources of information to put them into a numerical prognostic model. Recall that we're generating new information on primary productivity. We're generating new information on the taxonomic and size class abundance of different groups. We're generating new information on the abundance and productivity of zooplankton, and with that, the trophic transfer efficiency. All of those things can feed into these complex models that William, and I should also acknowledge Virginie, who is his PhD student working on this, and Robin Forrest, the PFO. They use all of that information to really understand in a prognostic, predictive way how the ecosystem is functioning. And it's these kinds of predictions then that are what allow us to inform our understanding of what may happen in the future if, for example, we shift the surface temperature and drive a certain response of phytoplankton. So William and Virginie and others are still pretty deep in this, but I'm optimistic that the information that we're giving them is going to help. So I left this one blank partly because I ran out of time, but also partly because I want to signal to you that the future is somewhat open as far as I'm concerned. We have these tools that we and others have developed. I think they've given us a lot of insight, new insight, just over the last few years about how lower level plankton dynamics are evolving in the subarctic Pacific. I think that information is potentially very, very useful for a variety of applications in fishery science, whether we're talking about short-term stock forecasting or whether we're talking about longer-term numerical models. The coupling of that information between what the people at the Institute of Ocean Sciences consider their bailiwick, the oceanography, versus the fishery science, it's bringing those two things together that is really where I think we're going to get the insight. And I do feel bad that I haven't done enough of that in the past, but I'm here now, and that seems like a good start. So thank you.